Hello and welcome to the final set of slides for week 11 on learning and memory, plasticity in the adult brain. This is also the set of slides that will be associated with our reading for this week, which was the Zeri et al. Article on the pregnant rat moms and somatosensory cortex. And I think I'm gonna try to do this in one video. It's a relatively short set of slides, but we'll see how it goes. So I want to start with some classic studies. If we hadn't read the Zeri article, we probably would have read one of these old classic articles by Marion Diamond and Mark Rosenzweig and a couple of other authors, Kretsch and Bennett. And I think they were at UCLA. And they discovered this really interesting phenomenon. They raised rats in three different conditions. This is the isolated condition, where it's a single rat per cage, which is a common way of housing rats in laboratory settings. The standard condition, sometimes called the social condition, in which three rats, two to four rats are raised together in a single cage, which is actually preferable for the rats. They tend to be less stressed when they're with their brothers or sisters. And since food and water are available in large quantities, there's really not much competition in that situation. So this is also a common way of housing animals in a laboratory setting. And then in the enriched condition, in which, as you can see, an entire litter of rats was housed together and the cage was extra large. There were two levels to the cage. There was a ladder or some kind of ramp that would let the rats get from one level to another. And then another feature of this enriched condition is they would put different objects, the experimenters would put different objects in the cage a couple of times a week. And those objects were, they called them toys. I don't know, rats really played with them, but the rats would explore them, move them around, and just kind of be exposed to new things to interact with in their cage. Sometimes there were running wheels as well. And they did this, I think, for reasons that had to do with learning and memory experiments. But at some point, kind of accidentally, they they wanted to measure levels of acetylcholine in the cortex, and in order to account for, I mean, when you're measuring some chemical in the brain, it's pretty common to then divide by the weight of the brain or the mass of the brain, so that you can standardize across animals who might be have different sized brains because they're different ages. And when they did this, they made the surprising discovery that the brains of the animals, at least it was surprising to them, the brains of animals raised in this condition were different than the brains of animals raised in the other two conditions in a number of ways, including the thickness of the cerebral cortex was larger in the enriched conditioned animals. And the ratio of the cortex to the subcortex was different in the animals raised in the enriched condition. And I say it was surprising to them, and it was surprising to the scientific community at the time. But maybe it shouldn't have been, because even Charles Darwin recognized that rabbits raised in captivity had smaller brains than rabbits that were caught in the wild. But to the scientific community of the 1960s, when these findings were first published, it was surprising, because at that time it was thought that you don't get new neurons in adulthood, and that any changes in your brain are really very small changes, and they're not changes in the structure of the brain. So how could one brain be larger? It was thought that the brain isn't like a muscle. Yeah, the more you work your muscles out, the bigger your muscles get. But it didn't seem like that was true for the brain, that the more you work your brain out, the larger your cerebral cortex is going to get. And so, in fact, when they published these findings, there was a lot of resistance to these findings. And so, over the course of 3 or 4 years, they replicated this study like a dozen times in publications, attempting to meet all of the various concerns that had been raised. You know, when somebody publishes something that doesn't make sense to you, brains getting bigger because you put toys in their cage, you read the paper very carefully and you try to find flaws or reasons that might provide alternative explanations for the findings. And so many of those were, I mean, that's the way science works. You want to make sure that the findings are as robust as they can be. And so many of those complaints might have been quite legitimate. But in addressing those concerns, the main phenomenon would be replicated again and again. And in fact, I think Rosenzweig at one point said that the phenomenon was so robust that he could do that in an undergraduate lab class. Um, he would teach physiological psychology just like I am. They would raise rats in different conditions. They would then kill the rats, take the cortex out, weigh the cortex, and the undergraduates with very little experience in how to handle anatomical tissue could tell the difference. I mean, it was that robust a phenomenon that it served as a college lab experience for a while in Rosenzweig's classes. So this phenomenon eventually was proven beyond a shadow of a doubt and came to be believed, and it helped to build this new idea about brain function, which we talked about in a previous set of slides. That yes, your genes influence your brain structure, and your brain structure influences your behavior, but then your behavior can also influence brain structure and can also influence gene expression. So the animals playing with these toys and being in an environment that created more sensory motor stimuli for them to interact with, that also is a thing that can influence the structure of the brain. Now, one of the things that that the researchers next addressed was, why is the cortex getting bigger? There's any number of reasons why the cortex could get thicker and heavier. One thing could just be that, I mean, when you exercise, so these animals are moving around more. When you exercise, your blood vessels get bigger. So maybe it was just, blood vessels getting bigger. Or maybe it was glial cells multiplying for some reason. It would be more interesting if there were changes in neurons. And it turns out it was changes in neurons. This was before neurogenesis was understood. And these guys didn't see any increase in number of neurons, but they did see increases in the size of the neurons and the size of the dendritic branches of those neurons. So the gray matter did increase in the cortex, especially in the occipital lobe, but also in other parts of the cerebral cortex. And so that was Marion Diamond was the anatomist of that group, and she did some beautiful studies looking at changes in neurons, individual neurons, as a function of being in this enriched condition. Now, probably the most dramatic finding by this group was that while their first studies were done on young animals, so they were, rats live about 2 years in captivity, maybe a little bit longer than they do in the wild, but they grow very quickly, and so they're sexually mature by about, certainly in 2 months, they're sexually mature. And in the first month, they are not eating solid food, or they don't wean until about 1 month. So at 1 month they wean, so they start exclusively eating solid food at 1 month, and then by 2 months they're adults. So that's a pretty accelerated time course. And a lot of these experiments were done in the post-weaning period. So you took 30-day-old rats, you put them in these enriched conditions, and then when they're adults, you would look at the brain, something like that. And so that wasn't maybe too surprising that a young animal might show these sorts of changes in neurons. But then they started to explore, what happens if you take adult animals that have been raised in standard conditions, and then once they become adults, you shift them into the enriched condition, or you shift them into the isolated condition, because all of these groups were different from one another. These had the biggest brains, these had the next biggest brains, and these had the smallest brains. And there were significant differences at each of these levels. So what happens if you switch them from one to another during the adult period? And that's what this figure is showing you. This is what the neurons look like, or an average neuron might look like in cerebral cortex in an animal raised in the standard conditions with 3 animals per cage. And if you move them into the enriched conditions, the the dendritic branches become even thicker and the neurons become even more complex. If you switch them into the isolated condition, the neurons become less complex over time and they start to withdraw their dendritic branches as these animals are stagnating. So it turned out that these changes were not limited to animals raised during the rearing period, but actually could occur in adulthood as well, which really this was the genesis of a lot of the stuff that you see now with like play classical music to your kids and those little leap pad things where you give kids these little books that talk to them and enrolling your kids in pre-K and these messages about how it's really important to enrich your children, or the motivation also for activity programs at senior citizen homes to keep their brains healthy, keep people being stimulated. These websites where you go and you play games and the websites claim that you're maintaining the health of your brain. Now there's a lot of pseudoscience built around this, so I don't believe in these websites where you go and train your brain, but I do believe in activity programs at senior citizen homes, and I do believe that when you're raising your kids, exposing them to a wider range of sensory motor opportunities is important. And I do believe that in adulthood, people who are engaged actively in social interactions or in learning, and there's evidence for this too. It's not just my guess. This app, this study is 70 years old or 60 years old now, so there's been a lot of time to pursue this very interesting area of research. And the weight of the evidence is that yes, if you stay socially engaged, if you keep your brain working, crossword puzzles or learning new material, you will live longer, your brain will stay healthier, you will stave off Alzheimer's disease longer, you will be less affected by minor strokes and things like that as you get older. I definitely believe in that. I tend not to believe in, I think that you get that best from social engagement and learning a range of things as opposed to doing one thing, like going to a website and playing games or something like that, or listening to classical music or something like that. Another point I should make about this before moving on is that the experimenters called this the enriched condition and this the standard condition, but that's all relative to a lab environment. If you instead compare these to a rat in the wild, then even the enriched condition is still a cage, and it's still a new toy every couple of days, as opposed to an animal in the wild that is facing changing conditions all the time. So you really could view these all as impoverished conditions, just less and less and less enriched as you go down. So some people viewed this as, here's all the advantages to your brain of enrichment. Some people viewed this as, here's all the disadvantages of your brain of not having enrichment. And that's probably closer to the truth. Now, there have been studies that compare even this condition to a wild condition. And we should keep in mind that rats always live longer in captivity than they do in the wild, because the wild is a dangerous place, especially for a small little species like a rat. But it is an important perspective to remember that all laboratory conditions are impoverished relative to wild conditions when it comes to sensory motor. And so when we think about humans and what will generalize to humans, the truth is humans, compared to rats raised in cages, have enriched lives. So the question is there something that we should do or could do to gain even more benefits than just living our regular everyday lives? And there seems to be some advantage to that, especially when you're dealing with clinical populations, right? So kids raised in orphanages versus just any kind of natural and normal environment where you're not in an institutionalized setting, or kids who are sick who have to spend a lot of times in a hospital. Or gosh, maybe even situations like right now where kids are on lockdown. And in situations on the other end of the life spectrum where you're maybe in a retirement community versus a um, what's the word I'm looking for? Um, some kind of assisted living situation. There, the differences, the gains might be pretty substantial if you have some activity program or some social interaction than if you are maybe in that situation and nobody comes to visit you because everybody lives far away or you don't have any living relatives or something. Then you're dealing with a big contrast between what's possible at that age in that state of health, and therefore the benefits you could have for the central nervous system versus in that kind of isolated situation. Now, those Rosenzweig studies, Rosenzweig, Diamond, Kretsch, and Bennett, led to a lot of interest in, well, what kind of changes are possible in the central nervous system in adulthood? And a lot of those studies of so-called adult plasticity have taken place in the somatosensory system because somatosensory cortex is easy to get to. It's right on the surface of the brain, and it's highly organized. Now, we haven't talked about the somatosensory system and won't in any great detail, so let me talk a little bit about somatosensory cortex. It's on the postcentral gyrus. It's right behind the central sulcus. And we've talked about primary motor cortex. It's kind of got a very similar organization, except that its somatotopy is much finer. So if you remember when we talked about primary motor cortex, we said that primary motor cortex is organized more around movements and goals of movements than around muscles and body parts. Somatosensory cortex is very much organized around body parts, regions of skin. Areas at the top of somatosensory cortex, the dorsal part of somatosensory cortex, receive touch information from the lower limbs on the opposite side of the body. So here we're on the left side of the brain. So this region's receiving touch information from the right leg. And if I touched my right leg, I could record electrical activity of neurons up in this part of somatosensory cortex. And if I stimulated that part of primary somatosensory cortex, like Penfield did in his experiments with or on neurosurgery patients, I would feel tingles on my skin on my right leg. And you can see that there are regions of skin associated, or regions of somatosensory cortex associated with the torso, the arm, individual fingers of the hand, the face, and parts of the oral cavity down here in the ventral part of somatosensory cortex. So it has this highly detailed organization. So much so that, as I said before, you can get down to an individual digit on the finger, on the hand, that's associated with specific pools of neurons in primary somatosensory cortex. I'm guessing here that, but this is roughly what the organization might look like. And that makes it very possible then to detect changes that occur in somatosensory cortex. So one of the questions that was asked back in the 1980s was, what would happen if a person suffered an amputation or nerve injury to the middle finger? And in fact, you won't probably quite be able to see this. Maybe you can see it, but I have a scar on my thumb. I have a scar on my thumb because about 12 years ago or so, I was carving a pumpkin on Halloween in the morning, and the knife slipped and it sliced through my thumb. And I looked down and I could see white, and that kind of scared me. I didn't know if I was looking at bone or what I was looking at. It was a really deep cut. And so I went to the hospital, and I had severed this tendon right here, which pulls your thumb back. And the nerve runs with that tendon. So I was also numb on the tip of my thumb. And the surgeon had to stitch the tendon back together to allow it to heal. And the nerve also will eventually heal. And I eventually got touch sensation back to the tip of my thumb, and I'm able to do this again. So I can say to the surgeon, I can do it. thumbs up. So the question would be then, well, if this nerve is cut, or if I lose the whole digit, then does the neurons here, they won't receive any input anymore. What happens to those neurons? Now, in the experiment I'm going to tell you about, let's imagine that the middle finger has that problem, right? It's either been amputated, it's been lost as part of an injury, or the nerve leading to that or bringing information from that digit has been severed. What happens to that? And that experiment has been done in monkeys, and then you can go back and you can record from those neurons, and these neurons here start to begin to receive touch information from the index finger and the ring finger, the neighboring fingers, which, if you think about it, a lot of the things that we do with our middle finger, we also do with our index finger and our ring finger. We're gripping things with our whole hand. And so having sensitivity, maybe extra sensitivity to those fingers, helps to compensate for the loss of sensitivity from the finger that's been injured. So it's a plastic change that actually helps you do most of the things. About the only thing that we do with our middle finger that we don't do with the neighboring fingers is show somebody that we're disappointed that they cut us off in traffic. That you wouldn't be able to do with this change. But any kind of gripping movements involving the whole hand, having extra sensitivity to the neighboring fingers is probably going to help you execute that behavior. What happens though if, like, my nerve grew back? What if we cut the nerve to the middle finger and then eventually the nerve to the middle finger grows back? Now we have no neurons in central nervous system that are representing that finger. Well, over time, and actually relatively rapidly, sensitivity to that finger does in fact return. So that's another plastic change that is going to help us after an injury. What about the opposite situation? The opposite situation is probably more common and certainly more devastating in people. What if there's an injury to the brain and not an injury to the peripheral tissue? Like there's a stroke, for example. What happens if there's a stroke involving, let's say, the middle finger? Then same thing's going to happen. You're going to be numb. But now in this case, the reason you're numb isn't because the nerve's damaged. You still have a good nerve and sensory receptors. That information's going to the brain, but it's going to a part of the brain that's not there anymore because of the stroke. It'd be nice if you could restore sensitivity to that finger since it still exists and the sensory receptors still exist. And they did this experiment with monkeys too. In fact, Zaria was part of this experiment, the author that you read on the rat pregnant mom paper. And they had this task where the monkeys, in order to get food pellets, had to fish them out of a well that they had to reach down into. And so they really had to have good sensitivity to a couple of fingers. And then they created a stroke. They lesioned that part of the brain that controls those fingers, and then the monkeys couldn't get the food anymore because they just had very poor sensitivity to the fingers that were registered by that part of the brain that had been destroyed. But over time, the monkeys got better and better at fishing the food out with those fingers, and also over time, when they looked, they found that healthy areas of brain that had not been damaged by the lesion began to represent the fingers that had no representation in the central nervous system. And the monkeys working with those fingers was a big part of causing those changes in the central nervous system. And that's related to things that we've talked about already. Eye patch therapy for damage to an eye, and constraint-induced movement therapy, which is the stroke model where we saw a video of a woman working with a hand that had been compromised by the stroke. Monkeys are doing the same thing here, basically. In fact, this study really helped to increase the use of constraint-induced movement therapy with humans, because we had the evidence that if you work that hand, you will get changes in the central nervous system that enable you to adapt and overcome the permanent loss of neurons that occurs from the stroke. So, given how important the use of the hand is in driving plasticity, um, all the studies I've told you about, there's been some injury and then some change in the brain. But maybe you don't need the injury to cause changes in the brain. Maybe what's causing changes in the brain is changes in how you use parts of your body. So maybe any change that you do with how you use your hands, you learn a new skill like typing, like playing the guitar, whatever it might be, maybe that's enough to cause changes in the organization of somatosensory cortex. And so that led to the study that you read. And I guess the question was like, how do we test this? How do we you could train a monkey to do a skill like typing or playing the guitar. That would be really cool. Um, but the one thing I really like about this paper is they focused in on something that really didn't require training. It was a natural behavior that was, and it was over a very short time period. Instead of training a complex behavior in a monkey, you're just using a natural behavior that occurs in these rats. And they focused on a part of somatosensory cortex that is very small. You do not have a whole lot of representation of the skin of the trunk region of your body compared to, like, look at all this space in somatosensory cortex devoted to touch information from the thumb compared to the amount of space devoted to trunk touch information from the trunk. The thumb's tiny, the trunk's huge, but you have more neurons devoted to moving to touch from your thumb than to touch from your skin of your belly. And that reminds me of another thing that I'm sure I've mentioned this semester, which is the more neurons you have dedicated to something, the better you are at that thing. And our touch sensitivity from our fingers and thumb is very fine. Our touch sensitivity from the skin of our back and our belly is not very fine. So if we want to explore a material, say, oh, let me feel what this feels like, we use our fingers to do it because that's where we have our best sensitivity. We don't go like this because we just don't have our back. We just don't have very good sensitivity in that part of our body. And that's been verified in psychophysical experiments too. So they focus in on this part of the body that has very little representation. And then they say, how can we make this part of the body get more stimulation? Because it's getting stimulation that drives changes in the brain, right? Using your hands, the monkeys using their hands to get the food pellets out, drove those changes after a stroke. How can we train some skill of the trunk or increase the amount of somatosensory input on that part of the body? Rats don't use that part of the body either. They use their whiskers and their paws when they're exploring. They don't use their bellies. But there's one time in a rat's life when the belly region gets stimulated a lot, and that's during the nursing period. So after a rat gets birth, they have, as you can see, anywhere between 5 and 11 or 12 pups. That's another difference, by the way, between wild animals and rats raised in captivity is the main reason that, if you think of a breeding facility that makes rats to be distributed for scientific experiments, they are obviously just artificially selecting their rats for how many pups they have. The more pups they have, the more those rats will be used in breeding colonies. And so, point is, in captivity, and also in captivity, you have better nutrition and all those sorts of things that foster large litters. So you tend to get a lot larger litters in breeding colonies than you would out in the wild. And so these rats tend to have a lot of pups, 8, 10, 12 pups. And when rats are first born, you can see they're very primitive looking. They're very altricial, to use a technical term. They have no fur. They can't thermoregulate. They can barely feed themselves. They have to huddle together for warmth. And the mom is in charge of the nursing episodes. The pups don't even, they kind of get kicked off the nipple when they're done drinking. And that's all up to the mom. And so the mom's got and mom guides them to the nipples. the mom's got to have very good sensitivity in order to um, govern those interactions. And they're always crawling around there, they're always underneath her. Um, and so that part of the body suddenly gets a whole lot of stimulation until about these pups here are probably about 20 days old or so, 25 days old. So now they're moving toward weaning and they're going to get a lot. So over the first 3 weeks is when there's a lot of somatosensory stimulation of that part. And I'm just going to show in this lecture one figure from the paper, but we'll go more into detail on it on Tuesday night and talk about the other features of this paper. But basically what they're doing is they're taking these rats and they are going into somatosensory cortex of the moms and they're mapping it out. So each dark spot here, this is the surface of the brain. You can see a blood vessel on the surface of the brain in the somatosensory cortex region. And each dot is where they experimenters have put in an electrode one at a time. So these recording sessions take 10, 12 hours. The electrode goes in, and then they have to touch every part of the rat's body to find out what sets off these neurons here. And once they've decided, they record it. So like this, these neurons here responded to touch to one of the hind limbs, the hind limb on the opposite side of the body. This spot right here responded to touch to the tail. And all of these spots inside this circle here responded to the touch of the ventrum, which is where the rat pups were crawling around, or I should say would have been, because this is a non-lactating rat. This is a rat that gave birth but never ended up with any pups. And this is a lactating rat. This is one that gave birth and was nursing for about 3 weeks. And then they looked in somatosensory cortex. And if you compare this control animal to this experimental animal, you can see that the amount of space devoted to touch information from this part of the skin is much larger, about 50 to 100% larger in the nursing moms than in the non-nursing moms, or in the rats, the female rats that didn't give birth at all. And so the comparison between these two is very interesting, because it's not like every rat that gets pregnant has these changes. It's only rats that are nursing, that are getting the somatosensory stimulation of that part of the skin, that show those changes in somatosensory cortex. And there are a number of other things that they document in this paper that we'll go into on Tuesday night. The bottom line from this paper is that if one part of your body suddenly gets more somatosensory stimulation, your brain will change in such a way to represent that touch information with higher accuracy. So I said that we don't have very good tactile resolution in this part of our bodies, but I suppose if we practiced every day, that's a pencil. That's a mouse, computer mouse, that we get better and better over time. And the reason we're getting better and better is because our brains are changing in such a way to better represent that information. As I said, we'll go more into that later. Now, this has also been shown in humans with more interesting skills than, well, I should say, in humans. I mean, it's been, I don't know if that nursing moms study has been replicated in humans, but certainly it is sort of cultural wisdom that when women, human mothers, are nursing, they talk about having higher sensitivity in that part of their bodies. And so, it probably is a similar phenomenon going on. But as I said, in other types of skills, so one of the interesting studies shows increased cortical representation of the fingers of left-handed string players. When you're playing a stringed instrument, if you're right-handed, which most of us are, you fret the instrument with your left hand. So this is the one where you need very fine control of your fingers on the instrument, the guitar or the violin, or the cello or whatever, in order to properly fret the instrument. And you have to be making very fast decisions with this and quick response to touch feedback. And so for stringed instrument players, you can show greater representation of the fingers of the fretting hand than the strumming hand or the bowing hand of the instrument. And you can also show that musicians have greater representation than non-musicians do. So both types of controls have been shown. So again, your brain undergoes changes in response to skill learning. All right, so that's it, and I think we talked in the previous lecture too about how people who are blind from birth or deaf from birth also show changes in cortex to help them better represent their surviving sensory systems. All right, so that's the end of this week. We'll look forward to the discussion of the Zeri paper on Tuesday night.